Guys, let's quickly talk about the role of sodium iodoacetate in sample preservation. All right, basically, sodium iodoacetate is used to preserve some samples in the lab, but basically, most students don't really know how sodium iodoacetate acts. Okay, and that's the role. That's like the mechanism of action. And knowing about how a sample works, or knowing about how a substance work is the biggest authority you can have, okay, because you actually know how the substance actually work. Okay, so on that basis we'll be talking about sodium iodoacetate. Basically we'll be talking about its role in sample preservation. All right, so this is how what sodium iodoacetate will look like. Okay, so sodium iodoacetate is an effective anti-glycolytic agent. If they say anti-glycolytic agent, that means what? It is against glycolysis. Okay, so to preserve glucose. Are you reasoning with me? Okay, so but it has no effect on urease though. All right, it may inhibit creatine kinase, but no significant effect on other chemical laboratory tests. Okay, so what is its mechanism of action? It acts by inhibiting glyceride 3-phosphate dehydrogenase. Okay, and this is an important enzyme in the glycolytic pathway. Okay, so when it inhibits this glyceride 3-phosphate dehydrogenase, now the pathway for glycolysis will not be able to proceed. Okay, thereby preserving glucose. Okay, if you see this. All right, so what is the uses now? If that is the mechanism of action, what is the uses now? Since it inhibits glycolytic pathway, it is used in preventing glucose loss during transportation as an alternative to what? Sodium fluoride. Okay, it is also used in treatment of what? Several muscle disorders, as it inhibits what? Creatine kinase. All right, so that's just it about the sodium iodoacetate, okay, and its role in sample preservation. All right, so see you guys pretty soon now.